Electrophoresis is one of the most common techniques for investigating DNA. It allows us to separate DNA fragments according to length. It is a separation technique based on the movement of charged molecules in an electric field. As you know, DNA consists of two sugar phosphate backbones, joined by complementary base pairs. The sugars are outlined here, and the phosphate groups are outlined here. The phosphate groups are negatively charged. Therefore, in an electric field, DNA molecules are attracted towards the positive electrode, the anode. Since the charge-to-mass ratio of DNA is uniform throughout its length, all DNA fragments are attracted equally towards the anode. To separate the fragments by length, a gel matrix is required. A gel matrix is a cross-linked substance that acts like a molecular sieve. For DNA electrophoresis, the gel matrix is a polysaccharide called agarose. The purpose of the agarose is to impede the migration of the DNA molecules. The larger the DNA fragment, the more difficult its passage through the gel. Smaller DNA fragments travel through the agarose matrix easily. Therefore, in a given time, small DNA fragments travel farther than larger DNA molecules. In practice, agarose gel electrophoresis is performed as follows. The gel is made by dissolving agarose powder in a buffer with a high ion concentration, high salt buffer. The liquid mixture is poured into a mold and allowed to set. A comb is added, as shown here. When the gel is set, the comb is removed and the gel is taken out of the mold. The comb has created small empty rectangular chambers called wells at one end of the gel. The wells will keep the DNA samples apart so that they don't mix with each other. The gel is submerged completely in high salt buffer in an electrophoresis tank. The high ion concentration in the buffer and in the gel itself will result in the arrangement being an electrical conductor. Each DNA sample to be analysed is mixed with a loading buffer containing glycerol and a blue tracker dye. The purpose of the glycerol is to make the samples dense so that they sink to the bottoms of the wells. The purpose of the blue dye at this point is to make the samples easy to see. To determine the length of the DNA fragments being analysed, a sample containing DNA fragments of known length is loaded into one of the wells. A lid is placed over the electrophoresis tank before the tank is plugged into a power pack and the electric current is switched on. Negatively charged ions travel from the cathode to the anode through the buffer and the gel, and the negatively charged DNA molecules migrate through the gel matrix in the same direction. Safety alert: never touch a liquid while there is an electric current running through it. The DNA is not visible to the naked eye. Bromophenol blue can be seen migrating through the gel and enables us to keep track of what is happening. The dye, being very small, will move faster than the DNA. When the dye approaches the end of the gel, the current is switched off. If the current was left running for too long, the DNA would eventually run off the end of the gel into the surrounding buffer. The gel is now removed from the electrophoresis tank and viewed under UV light. A fluorescent dye included in the agarose gel makes the DNA fluoresce under UV light. We can now see the DNA as discrete bands. Each band consists of DNA of a particular length. The lane containing the length standards will have many bands. For this reason, the length standards are often called the DNA ladder. The length of DNA molecules is written in terms of the number of base pairs, BP, or thousands of base pairs, kilobases, kb. By comparing the distance migrated by a DNA band in a sample to the distance migrated by the DNA bands in the DNA ladder, we can work out the approximate length of any DNA fragment. Agarose gel electrophoresis is used to check the success of procedures such as restriction digests, where DNA is cut into specific fragments, PCR, where multiple copies are made of a desired fragment. If a particular fragment is needed for further experiments, a DNA band can be cut out of an agarose gel and purified for further use. The number and size of bands can give us information about a DNA molecule. For example, suppose that a DNA fragment is cut with a restriction enzyme. A restriction enzyme is an enzyme that cuts DNA at specific base sequences to give a mixture of fragments referred to as a digest. A sample of the digest can be analysed by agarose electrophoresis as shown here. In lane 1, we have the DNA ladder. In lane 2, the single band of about 10 kilobases is a sample of DNA taken before the restriction enzyme was added. Lane 3 contains the digested DNA. We see 3 distinct bands, indicating that the enzyme cut the DNA into 3 fragments of different sizes. That suggests that the base sequence recognized by the enzyme occurred twice in the DNA fragment. Notice the approximate length of the 3 fragments. 5 kilobases, 3 kilobases, and 2 kilobases add up to a total of 10 kilobases, which is the same as the undigested intact DNA. Take-home points. Agarose gel electrophoresis separates DNA fragments according to length. DNA is negatively charged, so it moves towards the positive electrode when an electric field is applied. Smaller DNA fragments pass through the gel easily, so they migrate faster and further, i.e., distance moved is inversely proportional to length. When you look at an image of a gel, the larger DNA fragments will be seen as bands near the top of the gel, whereas smaller DNA fragments will be near the bottom. Comparison with the DNA length standards, the DNA ladder, reveals the approximate sizes of the various DNA fragments.